Good morning, everybody. Let me introduce myself. My name is Jean-Marc Bellemin. I'm the Chief Financial Officer of Iovance Biotherapeutics. I want to thank you for your time today to learn more about Iovance and our mission to be the global leader in innovating, developing, and delivering T cell therapy for patients with cancer. As a public company with many goals in front of us, I will be making forward-looking statements, and I want to point your attention toward this slide for a more in-depth description of the forward-looking statements. To begin, let me introduce to you our company. So Iovance is a late-stage oncology company developing potential one-time cell therapies that harness the body's own immune system to fight and kill tumors. We were founded in the Bay Area and have grown to position Iovance for success in 3 different locations. First, our headquarters is in San Carlos in the Bay Area. Then we have built out our own manufacturing facility called Iovance Cell Therapy Center, or ICTC, in Philadelphia, Navy Yard, which is the US hub of cell and gene therapy. Finally, we have a research facility located in Tampa, close to Moffitt, Orlando Health, and other key US oncology centers, where we have collaborated for many years. Outside the US, we have a European foothold in key location, which makes sense for our current clinical studies, and potential commercial activities in the future. Today, we have more than 270 employees with an average of 3.5 years of cell therapy experience across all of our locations. On the following slide, I would like to highlight our 4 areas where we believe we may create significant value for patients, collaborators, investors, and other key stakeholders. First is our platform. We are leveraging a technology called Tumor infiltrating lymphocyte, or TIL, which was initially proven by Dr. Steve Rosenberg at National Cancer Institute. We have transformed his lengthy academic process into a scalable and commercially viable 22-day manufacturing process that has been used to treat more than 450 patients with a success rate north of 90%. Today, we are the latest stage TIL company, and we have built a strong intellectual property portfolio across all of our know-how from this proprietary TIL platform. Turning to our pipeline, our lead TIL programs in melanoma and cervical cancer are in pivotal program stage, with a potentially registration supporting program in non-small cell lung cancer, as well as additional clinical studies in head and neck, lung, and certain type of leukemia. Our strategy begins by identifying patient populations with defined unmet medical need, where we may pursue accelerated paths to initial approval with single-arm studies and overall response rate endpoints. As such, our TIL therapies have been granted several important designations from the US FDA, such as RMAT or BTB, that recognize the unmet needs we are addressing and the data that we have shown in the clinical to date. Beyond our initial indication, we are pursuing earlier lines of treatment for TIL in combination with approved therapy, which I will touch upon later. And we also have several collaborators doing exploratory studies in additional indication. In addition, we have the assets we believe we need to transition to our next stage of growth. Our current cash position leaves us well capitalized with a strong balance sheet to advance our clinical programs and prepare for commercialization. In addition, on our own Iovance manufacturing facility will support our internal clinical and commercial supply of these novel cell therapies. Through our leadership position in TIL and what we have learned to date, we have also been strategic in building our intellectual property portfolio to maintain our TIL leadership for many years to come. And finally, through our collaboration with the leading US academic cancer research centers in the world, we may explore access to TIL for additional patients in new indications to inform our future developments. We do have collaboration and or licenses to cutting-edge technologies, such as the talent platform from Cellectis to pursue the next generation of TIL approaches. Our pipeline is divided into multiple domains, as indicated here. The first area is TIL as monotherapy in patients who have received prior treatments. This is being studied in pivotal cohorts in patients with metastatic melanoma and advanced cervical cancer, as well as additional clinical studies in non-small cell lung cancer and head and neck cancer. We are also exploring the utility of TIL in combination with immune checkpoint inhibitors as an earlier line of intervention. We are exploring this combination in patients with the same tumor types as the monotherapy and have reported initial promising data in melanoma and head and neck. Third, Iovance continues to innovate through new products and manufacturing processes, including a peripheral blood lymphocyte, or PBL, therapy approach for blood cancers, genetically modified TIL, and a third-generation manufacturing process called Gen 3 that is now 16 days. So we have a lot going on, as you can see on the pipeline image. And so in my short presentation today, I will highlight our most recent clinical data set for TIL in melanoma and in non-small cell lung cancer. Let me first describe the high-level mode of action for TIL. So TIL is personalized to each patient as the person's own cells became their own therapy. Patient-specific immune cells called the TIL are removed from the tumor so that they can be amplified, rejuvenate outside the body, then infused back into the patient. TIL has a unique mechanism of action, or how it works inside the body. In a healthy individual, when cancerous cells are detected in the body, the TIL come to the site of the tumor, launch an attack. In cases where the disease does prevail, the TIL come to the site of the tumor, but the tumor has overpowered the TIL from successfully doing their job. Through a manufacturing process, which I will explain further in a moment, we produce billions of newly replicated T cells to return to the body and attack the tumor. This next slide highlights how TIL is a personalized and differentiated therapy that targets a diverse array of patient-specific cancers antigens, which are unique molecules on the surface of the tumor cells. Once the TIL have been infused, they enter the patient's bloodstream, migrate to the site of the tumor, guided by the chemical signals released by the tumor. When the TIL reach the tumor, the T cell receptors recognize the antigens and tumor lysis, or killing, is initiated. On this slide, we have more detail on the patient's journey on the manufacturing process from initial resection, from when tumor is removed to when it is administered back to the patient. So let me quickly describe it to you. We begin by having the physician identify the target tumor. They surgically remove approximately 1 centimeter of that tumor, which is very small. The tumor sample is shipped to our centralized manufacturing facility in Philly to undergo a 22-day manufacturing process to culture and expand the TILs, which is the flow as the bottom of the slide. We introduce specific ingredients in the culture that helps grow to at least 1 billion TIL cells. After we confirm specific criteria for the finished TIL product, we cryopreserve or freeze the TIL therapy and send it back to the patient. The TIL can be thawed and infused back into the patient whenever the authorized treatment center and the patient are ready. During the TIL therapy regimen, back up toward the top of the slide, we prepare the body to receive TIL through lymphodepletion to remove the hostile and microenvironment of the tumor, followed by TIL infusion. After the TIL is infused, a short course of interleukin-2 or IL-2 is administered, not as a therapeutic intervention, but to promote TIL cell proliferation inside the body. Once the immune system is back to normal, the patient is then discharged. Let me talk about ICTC. So internal manufacturing has been a key priority at Iovance in order to fully control quality and time of all our processes and also reduce future cost of goods. Our ICTC is a state-of-the-art manufacturing facility in the Philadelphia Navy Yard. We are preparing to supply clinical studies from our ICTC in the near future. We just announced, today, November 23rd, that we have supplied the first patient from our manufacturing. and we expect to provide commercial supply from this facility upon potential BLA approval. The current capacity may meet the demand for thousands of cancer patients. So let's talk about the solid tumor market. As mentioned, we have the potential to introduce the first cell therapy in a solid tumor indication. And this next slide demonstrates the clear unmet need and significant market opportunity across various solid tumors. CAR T-cell therapies have been successful in certain blood cancer, but 90% of all cancer cases, meaning 1.6 million patients in the US, are solid tumors, and there are currently no cell therapies on the market for solid tumors. Our lead TIL therapy, lifileucel, is being investigated in metastatic melanoma. Melanoma, as you can see on the slide, is a clear unmet need with more than 96,000 diagnosed patients and north of 7,000 deaths annually in the US alone. Unfortunately, patients who progressed after initial treatment have a low response rate to retreatment and typically short survival if you look at the published data. Lifileucel has shown promising clinical data for this patient who progressed after anti-PD-1 therapy. Available care after checkpoint inhibitor is limited to chemotherapy, which is associated with poor outcome. And we have an overall response rate of only 4 to 10% and overall survival of around 7 to 8 months. Lifileucel has also shown initial clinical data that suggests a potential additive effect in earlier line melanoma in combination with anti-PD-1 therapy. I will highlight both of these data sets next. So here we go. So data for lifileucel, both as a single agent and in combination with immune checkpoint inhibitors, were recently presented at the American Society of Clinical Oncology annual meeting. In the top panel on this slide, you can see the overall response rate of 36% for patients receiving single-agent lifileucel after progression on prior anti-PD-1 immune checkpoint inhibitors and BRAF and/or MEK inhibitors if they had the BRAF mutation. The median duration of response was not reached in this patient after a median follow-up of over 33 months. You can see in the waterfall plot in the upper right panel of the slide that tumor reduction was seen in most patients, 50 out of 62. And the best response according to objective response criteria or RECIST are color-coded as red for progressive disease, gray for stable disease, blue for partial response, and green for complete response. The downward arrows also indicate patients who have continued to have tumor reduction after prior evaluation, and those labeled with an asterisk have the BRAF mutation. The bottom panel shows our experience with lifileucel plus pembrolizumab, an anti-PD-1 immune checkpoint inhibitor, in patients who had never received an immune checkpoint inhibitor before. We saw a very encouraging response rate of 86%, and the waterfall plot for this patient showed 3 patients achieve a partial response, blue bar, 1 patient achieve a non-confirmed complete response in the hashed green bar, and 2 patients achieve a complete response, the solid green bar. Together, this promising data support continued investigation of lifileucel in various treatment settings in patients with metastatic melanoma. So let me pivot to non-small cell lung cancer indication. At Iovance, we are also very excited about the potential for TIL in non-small cell lung cancer. And earlier this year, we showed our first set of clinical data in this indication, which I will highlight in a moment. Lung cancer is one of the most common cancers, with more than 2 million new cases and 1.8 million deaths worldwide each year. Most patients with metastatic non-small cell lung cancer develop disease progression with first-line immune checkpoint inhibitors, with or without chemotherapy. Then, second-line treatment with docetaxel produces a low 9 to 13% overall response rate. Effective strategies to provide deep and durable responses are urgently needed for patients with checkpoint inhibitor treatment resistance. At Iovance, we are investigating TIL alone, following immune checkpoint inhibitors, and in combination with checkpoint inhibitors in our line of treatment. Let's look at some clinical data. The slide presented here showed the key highlight of the LN-145 data from 28 heavily pretreated patients with metastatic non-small cell lung cancer. After progression on at least 2 prior therapies, overall response rate in this patient was 21%, with a median duration of response not reached at 8.2 months of median study follow-up. You can see in the waterfall plot in the upper right panel that tumor reductions were seen in most evaluable patients, 19 out of 24, and the best response according to objective response criteria, or RECIST, are color-coded as red for progressive, gray for stable disease, blue for partial response, and green for complete response. In the lower right panel, you can see the time to response for the 6 patients we responded, which shows early and sometimes deepening responses in this patient. Together, these promising data support continued investigation of LN-145 in patients with metastatic non-small cell lung cancer. So beyond our clinical pipeline, we are committed to continuous innovation in cell therapy, including gene-edited cell therapy that may extend and improve life for patients with cancer. This includes expanding our TIL therapies into new solid tumor indication, further optimizing TIL for greater potency in existing and new indication, and also to streamlining manufacturing to deliver TIL to patients even sooner. From left to right on this slide, I can hit a few of the key highlights. We have a novel IL-2 analog license from Novartis, which may further optimize the overall TIL therapy regimen, which includes a short course of IL-2. We then have a selected and genetically modified TIL, including PD-1 selected TIL in certain Iovance studies and PD-1 inactivated TIL to potentially increase potency of our TIL products. And finally, our Gen 3 manufacturing process is 16 days and may further improve on our already fastest in the TIL industry process of 22 days. To summarize the presentation, we are pursuing large market opportunities with solid tumors with significant unmet medical need, beginning with TIL alone and moving into combination therapy in earlier line with several investigator-sponsored studies underway to explore new indication. We clearly have the potential to receive approval of the first ever one-time cell therapy in a solid tumor indication with promising clinical data and regulatory designation, such as RMAT and BTD, to facilitate more frequent dialogue with the FDA. And lastly, we are differentiated by being the first drug sponsor to scale up TIL manufacturing at a centralized facility to become a fully operational internal facility for clinical and commercial supply of a novel cell therapy. I want to thank you for your attention and wish you a great rest of the day.